Hi. First of all, I would like to thank the organizers for inviting me today to give really the regulatory insight into the non-clinical testing of human medicinal products, and in particular to the challenges and the opportunities that are related to the acceptance of 3R methods. And first, I would like, of course, with the usual disclaimer that the views that are expressed in this presentation are really my views and not the opinion of the Belgian Federal Agency for Medicines or of the European Medicines Agency. And I would like to start off this presentation by giving you an insight into the regulatory environment in the European Union. And we are talking about human medicinal products, so we are talking about the Human Medicinal Product Agency, and the EMA has published a regulatory science strategy which focuses on different regulatory science aspects up to 2025. And into the heart of this strategy is a core recommendation to the leverage and the qualification of 3R methods. EMA really wants to see more of these methods coming to the agency and start a discussion on the definition of qualification, on the definition of context of use, on endpoints and reference compounds. And the basic heart of all this discussion is to engage with stakeholders in order to create a communication channel and to establish really a good European regulatory network, for instance on organ-on-chip, which is a hot topic at the moment. Now, ethical use of animals has been in the heart of the activities of the EMA already since early 2012, with the creation of the joint 3Rs working party, which is a working party related to every aspect of 3Rs, both for human medicinal products and for veterinary medicinal products. So some of the activities up to 2017 have been published in a report, and I want to focus on two different aspects that were carried out with this working party. The first thing that we did was really to draft a guideline on the principles of regulatory acceptance of 3R testing approaches. And this guideline is applicable, again, both for human and for veterinary medicinal products. And it defines what is regulatory acceptance. And most of you might think that regulatory acceptance is only the incorporation of a new 3R testing approach into a regulatory guideline, but it is also on a case-by-case basis the acceptance by regulatory authorities of new approaches that are not yet incorporated into a testing guideline but are really used for regulatory decision-making. And this guideline goes on by stating different criteria for regulatory acceptance. And this is not rocket science, it's just having a defined test methodology like protocol and endpoint, establishing and defining the relevance within a particular context of use, including accuracy, and then definition of the context of use itself with the limitations. And I think that's also important. You have a method and inherently you also have limitations. And we're thinking also of reliability and robustness. And then finally, what was introduced also into this guideline is the voluntary submission of data. And this is the so-called safe harbor procedure, where data that are generated by using a 3R method or a novel method can be submitted together with the data that are generated with the usual methods in order to really underpin the qualification of this method and foster regulatory acceptance in this sense. Now, with this working party, we have also done a big job by revising and looking through all the guidelines that are related to animal use and see where there were already opportunities for 3R's implementation, but also where there could be in the future new opportunities for 3R implementation. And you see here a screenshot of the reflection paper for the human medicinal products, and there is a parallel one for veterinary medicinal products. And it encompasses both quality testing, non-clinical testing, product-specific guidelines like for biosimilars and advanced therapeutic medicinal products, and also the more traditional non-clinical testing for small molecules and biologicals. And I will take you through now the different 3Rs opportunities that were identified in the non-clinical testing requirements. And this starts off with the pharmacology part. And the first guideline that we have related to pharmacology is the ICH S7A, a safety pharmacology guideline for human pharmaceuticals. And this guideline recommends that in vivo studies are carried out to assess potential effects on the central nervous system, on the respiratory system, and on the cardiovascular system, with an additional guideline, the ICH S7B, that pertains to the detection of the potential for delayed ventricular repolarization events by human pharmaceuticals. Now, when we're thinking about 3R opportunities, what has been identified is the integration of endpoints in repeat dose toxicity studies, such as in the context of ICH S9, which is related to anti-cancer medicines in an advanced cancer setting. And this means that this obviates really the need for standalone safety pharmacology studies. And most importantly also, ICH S7B is currently under revision. A Q&A is being drafted, and this Q&A integrates also all the activities of the SIPA network in the US and focuses a lot on in vitro and in silico approaches for the detection of pro-arrhythmic effects. Okay, now moving on to pharmacokinetics. And pharmacokinetics is the general absorption, distribution, metabolism, and excretion studies, which are defined in ICH M3 and a number of additional explanatory guidelines. Now, the 3R opportunities that we can identify here are related really to in vitro work. A lot of standard in vitro models are encourage for the comparison of in vitro metabolism across the species to really pinpoint which are the relevant species from a pharmacokinetic standpoint. Also to look at effects on enzyme P450 activity, protein binding, absorption studies using CACO2 cells, and then also when we look at the clinical guideline for the investigation of drug-drug interactions, we see that there is really the statement that in vitro approaches are the preferred option here. And then also I want to draw your attention to the ICH S3A Q&A that really pinpoints that microsampling methods in toxicokinetic studies are encouraged to reduce or eliminate even the need for satellite groups to analyze toxicokinetics within repeat dose toxicity studies. Moving on to toxicology, and you see that you have a whole range of toxicity studies that can or have to appear in a non-clinical dossier, and I will take you through one by one. Now, start with the easy one, single-dose toxicity studies. The requirement for having single-dose toxicity studies has been obviated in 2010 following a Q&A on the withdrawal of a specific note for guidance on single-dose toxicity studies, and it has been agreed to remove this guideline and guideline ICH M3 that defines the type of non-clinical studies that have to be carried out and submitted to the regulatory authorities to support clinical development and marketing authorization, really defines the scientific basis for supporting this approach. Moving on to repeat dose toxicity, and it is clear that for repeat dose toxicity studies, we want studies in relevant animal species. Usually there is the need for a rodent and a non-rodent study in an appropriate duration, and the duration has to be in line with the duration of proposed therapeutic use in humans, and the administration route is the clinical route. Now, what are the 3R opportunities? One species is possible if clearly justified scientifically, and for this justification often a dialogue is encouraged with the regulatory authorities through scientific advice, which can be either at the national level or at an EMA level. Also, the inclusion of additional in vivo endpoints in order to reduce animal use, if scientifically justified, is really encouraged. We're thinking about safety pharmacology endpoints and genotoxicity. Reversibility then, it can be integrated only in the chronic toxicity study if possible. And then there are also a number of specific requirements for anti-cancer medicines and for biologicals. For anti-cancer pharmaceuticals, 3 months data are sufficient for marketing authorization, even if the clinical use is longer. Also, no non-rodent studies are used for cytotoxic pharmaceuticals, and there is no need for recovery groups based on scientific rationale. So you see, this is always coming back. Scientific rationale. Please think from a scientific standpoint, what is the non-clinical program for your product? Biologicals then, you only need 1 dose group and 1 control group if this is scientifically justified, and 1 relevant species can be sufficient for the chronic tox studies. And by definition, this should be the lowest phylogenetic species, and also similar tox findings from biologicals in the same class, and the findings should be understood from a mode of action. So again, scientific rationale. And reversibility should only be carried out in 1 justified dose level. Genotoxicity then. The guideline for genotoxicity was also revised not so long ago in regulatory terms then, and there are 2 options that are proposed into this guideline. The first option is quite the traditional option, going from an in vitro Ames assay and an in vitro clastogenicity assay supplemented by a standalone in vivo clastogenicity assay. However, option 2, which is now the preferred option, is integrated, which is a standalone in vitro Ames assay, and the in vivo endpoints for chromosomal damage within 2 different tissues are integrated in the repeat dose toxicity study. Carcinogenicity. And carcinogenicity, having been in discussions around animal use, are really contested studies. These are long-term rodent studies carried out in mouse and rats, 2-year studies, or a long-term rat study supplemented by a 6-month study in a genetically modified mouse. Now, what are the 3R opportunities here? I will start off by biologicals under ICH S6. There is no need for 2-year carc studies unless there is cause for concern. And the ICH S1 work then. Now, there has been a group set up numerous years ago to look at how to refine the approach on the need for carcinogenicity testing. And to make a long story short, in fact, a new testing paradigm has been under evaluation, which is based on a more comprehensive and integrated weight of evidence approach to address the risk of human carcinogenicity of small molecule pharmaceuticals, and to define, in fact, the conditions under which a 2-year rat carcinogenicity study could be omitted. So there are kind of 3 categories. One is the compound will certainly be not carcinogenic, based upon a weight of evidence, which comes from a range of studies, pharmacological studies, also looking at the outcome of repeat dose toxicity study, is the compound genotoxic or not, etc., etc. The second quite open, clear category is the compound will certainly be carcinogenic, and in both of these extreme categories, there is no need really to do a carc study. On the other hand, you have the gray zone with a lot of question marks, and there you might still need a carc study. So we're hoping that the implementation of this new weight of evidence approach will really reduce the number of animals used for carc studies. And recently an addendum to this guideline S1B on the testing of carcinogenicity of pharmaceuticals was published. It is currently still under public consultation, and I invite you to read it because comments can still be submitted until the 22nd of October of this year. Local tolerance then. Local tolerance testing is usually needed for products that are applied topically or are injected intravenously or intramuscularly and so on, or subcutaneously. Studies have to be done to look at irritation, and for products that are applied on the skin, to look at sensitization potential. What are the 3R's opportunities? And you see them here. Standalone animal studies are generally not required for looking at irritation. Endpoints can be integrated into the repeat dose toxicity studies. And then for sensitization potential, in vitro testing and integration of appropriate endpoints in repeat dose tox studies for irritation is highly recommended, but the last part I already mentioned to you. Now for sensitization, I want to come back to that because we know that a lot is going on around sensitization testing for cosmetics and for chemicals, and we really wanted, when we revised this guideline, to integrate already the possibility to submit these type of in vitro data to also the pharmaceutical regulatory authorities. Other tox studies, and there is a whole menu of tox studies that can be done, immunotox, antigenicity, and so on, and I want to focus here on immunotoxin dependence testing, impurities, and photo safety assessment. What are the 3R opportunities here? Dependence and immunotoxicity studies are only needed when standard non-clinical studies indicate a cause for concern. Again, weight of evidence. There has been a draft reflection paper on the qualification of non-genotoxic impurities. Why? Because if you need to qualify them, now it is said that you need to do a 2-week repeat dose toxicity study. And with this reflection paper, we wanted also, again, to introduce a more reflective approach, looking at read across, also looking at other parameters, in vitro testing, and so on, to try and qualify non-genotoxic impurities. And finally, for phototoxicity testing, it is really clear that we need a combination of photochemical evaluation and in vitro tests to have an idea already if there is a phototoxicity for the particular compound under study, and then only additional studies are needed on a weight of evidence approach. There is no need for photocarc studies, and there is also no need for photoallergy testing. Environmental risk assessment, also this guideline is under revision. Environmental risk assessment for human pharmaceuticals is related only to the use of the pharmaceutical and not the synthesis or the manufacture of the compound. 3R opportunities. The revision of the guideline includes, of course, an optimization of the testing strategy that is currently in there, and the methodology in view of the 3Rs. And then I come to the pièce de résistance, which is really the reproductive toxicology. Why is pièce de résistance? Because this guideline was very recently revised, and there was a lot of attention for 3Rs within the revision process, which took, by the way, numerous years. So the 3Rs opportunities are the following. There is a refinement of dose selection approaches in comparison with the approach that was initially in there. For embryo-fetal development testing, so the teratogenicity testing, under certain circumstances, it may not be necessary to conduct studies in multiple species. The circumstances are here underneath. For biologicals and anti-cancer products, also, again, there is a reduced package possible and specific approaches that allow a reduction of animal use in these particular contexts. And then I want to really focus the attention on in vitro methods. And into the guideline S5 and in the whole revision process, there was a lot of attention for the in vitro alternatives, ex vivo alternatives that are available for looking at teratogenicity, for the induction of malformation and embryo lethality. Now, instead of recommending only a or a number of particular in vitro or ex vivo tests, we decided to focus on defining the criteria for qualification of the methods in order to allow flexibility and technological progress, and as such to allow a whole range of in vitro methods for submission and for the detection of teratogenicity, more specifically malformation and embryo lethality. So it is stated into the guideline that the data that are generated from qualified alternative assays conducted either alone or in conjunction with one or more in vivo studies can be used to support not only hazard identification, and this is important, but also risk assessment, and this under limited circumstances. And the potential uses, I will not go through them specifically, but see that the context of use of the in vitro methods is really listed within this guideline and within the context of detecting malformation and embryo lethality. Also mention of GLP and qualification of context of use. And the guideline has a very elaborate annex, and in this annex all the qualification criteria are listed. So you see them here, and I will certainly not go through them in very much detail, but these are the proposed assay qualification information that needs to be provided to regulatory authorities for a new in vitro method that will be used to support hazard and identification and risk assessment in this context. And what are we seeing? We're looking at a thorough description of the predictive model. What are the endpoints? What are the species it predicts? We're looking at a discussion on positive and negatives in the assay. We're looking at a list of reference compounds, and I will come back to that. What is the performance of the test method over an appropriate range of biological and chemical domains in line with the context of use? If we are having a test battery or tier testing approach, a clear description of every separate assay has to be provided, and a global qualification of the prediction model, and then an expectation that, at least if you are using it for risk assessment, the assay should be conducted in accordance with good laboratory practices. And also, it should be stated if an 3R assay has already been submitted for qualification to another health authority, at least that we can also take this into consideration. And then I would also like to focus on the reference compounds. So into this guideline, a list of reference compounds is being provided, and you see the table. And I focused on only one reference compound. And sorry, I realize that this is a small table, and you can revisit it within the guideline and look at it at your own pace. But for every reference compound, it was ensured that we had sufficient data richness and homogeneous criteria were applied to select the reference compounds. So we needed to have data from rat and rabbit, and also to have an idea about the no observed adverse effect level, the lowest observed adverse effect level, and not only the doses, but also the plasma exposure levels that were associated with these parameters. And this is applied for every reference compound in the list. Moving on to technological progress, and we have heard in a lot of conferences since numerous years about organ-on-chip technologies, body-on-chip technologies, and so on. And really, this is an area where regulatory science has to keep pace with technological development, and we have to evolve to a mechanistic-based safety and efficacy testing. And for this, there is only the need for interaction between multiple stakeholders in order to ensure that these methods are qualified and fit for purpose, and that science-driven and mechanism-based strategies are delivered and are available to both end users but also to regulatory authorities. So in order to keep in pace with the development, a first workshop was organized in 2017 within the European Medicines Agency on non-animal approaches in support of medicinal product development, and what are the challenges and opportunities for the use of microphysiological systems or organ-on-chips. So this workshop aimed to map the current state of the science, to develop a common understanding of the benefits and limitations of MPS systems, identify the gaps in safety testing, and stimulate the research using MPS to address gaps and to really encourage the use of regulatory testing. And mainly also this first workshop had the intention to foster the dialogue between the developers that were present and the end users that were present and the regulatory authorities, and hence to facilitate regulatory acceptance. So everything was published in a workshop report, which you can find on the website of the EMA. And I just want to focus now on what are the action points that came out of this 2017 workshop. And there were two breakout groups during the workshop, and hence the action points are defined under these two breakout group headings. The first one is related to the criteria for regulatory acceptance, and there really it was focused that collaboration is needed. Collaboration to define guidance for method developers on qualification, what are the expectations of the regulatory authorities, and what are the expectations also of the method developers and the end users. Looking at endpoint-specific performance standards and reference compounds, we really need that, per organ system and per endpoint, of course. We have to agree on a kind of stepwise approach for organ-on-chip technologies. How will we proceed with regards to qualification and with regards to regulatory acceptance? Start simple, go more complex maybe. And discuss also on the applicability of clinical biomarkers, the whole reflection around gold standards, and identify the degree of flexibility in order to allow for continuous application of qualification criteria in order to avoid reinventing the wheel every time we get a new model. Progress towards regulatory acceptance, and this repeats a bit the parts that we already addressed. Discussion and agreements are needed on endpoints by all stakeholders. There is a need to have more precise regulatory guidelines in consultation with all stakeholders. Regulators need to be consulted to define the gold standard for each endpoint and system. And we really want to focus on human-based systems to have clinical data as well. And data sharing is a key for progress. This is possible through the process of method qualification under the safe harbor within the EMA, but maybe other processes are also possible. But the bottom line is communication between all stakeholders and involvement of regulators. Just to have an idea of context of use for these technologies, and this is a table that comes out of the ORCID project, which is a European project that wanted to define a roadmap for acceptance of these organ-on-chip technologies. And here, so the ORCID project evolved towards the European Organization for Organ-on-Chip. And a lot of workshops were organized and different contexts of use were defined for these organ-on-chip technologies, going from disease mechanisms, drug efficacy, drug toxicity testing, but also up to the very complex area of personalized medicines. And every type of context of use will have its own inherent extent of qualification that needs to be defined. Wrapping up, what are the take-home messages from this whole reflection? With regards to regulatory acceptance of 3 Rs, collaboration is really key. We need to include regulators upfront in every new method developer, because the new methods really need to answer regulatory questions if you want to at least to apply them in that context. And the regulators can then provide also insight in the tools that are needed, or what are the gaps within regulatory science that need still a method to answer them. What are then the specific areas for regulatory input? We can provide assistance in qualification criteria and context of use. And I have been talking a lot about European context, but this is a global matter. And certainly if we're thinking about organ-on-chip technologies, we need to collaborate also between regulatory authorities globally. A discussion on reference compounds. We need to avoid having millions of reference compounds lists for the same endpoints. Harmonization of reference compounds should be possible, and also with an eye for data richness. And then performance standards per endpoint, per model should be defined, and as such that method developers can know what really are the expectations for qualification. In this whole setting, training is important, and training not only of end users and of method developers, but also of regulators. We need to know what we're dealing with, and hence training of regulators is important. And I just wanted also to highlight the whole roadmap that was developed for organ-on-chip in Europe, which encompasses also a lot of attention for method validation and qualification. Second part, and what this is really related to the submission of data to the regulatory authorities. In this context, the European Medicines Agency has a number of procedures and entry ports for data discussion. One of this is the more formal way of qualification. Qualification of novel testing approaches, there is a guideline for that on the website of the EMA, which really describes the procedure, the email addresses and so on, how to go about. And the other part is the EMA Innovation Task Force. This is really free of cost, important, EMA platform for an early dialogue and an exchange of information on non-clinical methods and models. And when you come to the Innovation Task Force with a novel methods and want to move towards qualification, but you're not sure how, or you want to brainstorm a little bit, a discussion is possible within the agency with the relevant experts for the methods. And with that, I would like to really thank you for your attention, and I hope to have given an insight into our position. And if you have additional questions or you want to know more, please, here are my contact details, or you can also contact the EMA within the Innovation Task Force or our national agency, which also has an innovation platform. Thanks a lot. Bye.